A very good day to all of you children. Today, let's continue our lesson on molecular basis of genetic diseases. The other day, we discussed about three major types of genetic diseases. Can you remember them? They are the chromosomal diseases, monogenic diseases and polygenic diseases. Among these genetic diseases, most of the monogenic diseases are inherited according to the principles of Mendelian inheritance. We can find almost all the monogenic disorders with a Mendelian inheritance so far identified in this database. That means we can easily find the diseases with the Mendelian inheritance and the genes which are mutated in these diseases and also the types of mutations on those genes using this database. For example, suppose that you want to have information about the disease amelogenesis imperfecta. Can you remember what amelogenesis imperfecta is? It is a monogenic disease affecting the enamel of our teeth. When you want to know about the inheritance pattern of this disease, what you should do is type the name of this disease in this box. Then the OMIM database will provide all the necessary information about this disease to you. This information will include the names of the mutated genes and the location of these genes on chromosomes and also the type of inheritance. Here AD stands for autosomal dominant and AR stands for autosomal recessive and XL stands for X-linked inheritance. Well, before we discuss about these inheritance patterns, let's try to recall our knowledge on genes and alleles of our chromosomes. As you all know, our nuclear genome contains 22 pairs of autosomes and a single pair of sex chromosomes. If we look at our first chromosome, it has 2 copies. We inherit one of these copies from our mother and the other one from our father. It is the same for the rest of the chromosomes also. Since we have 2 copies for the same chromosome, we have 2 copies for the same gene also. Different versions of the same genes are called alleles, and they are found on the same position of the homologous chromosomes, and these are called the loci of that gene. For example, this is the locus of the gene A, this is the locus of the gene B, and this is the locus of the gene D. If the 2 alleles are identical, we say that they are homozygous. If the alleles are different, they are said to be heterozygous. Physical characteristic resulting from expression of an allele is called the phenotype. In this case, brown eyes is the phenotype, and in this case, blue eyes is the phenotype. Allele of a gene responsible for a physical characteristic is called the genotype. In this case, capital B is the genotype responsible for brown eyes phenotype. In this case, capital B simple B genotype is responsible for the brown eyes phenotype. In this case, simple B genotype is responsible for the blue eyes phenotype. And usually, dominant alleles determine the phenotype both in the heterozygous state and the homozygous state. And the recessive alleles will determine the phenotype only in the homozygous state. Because when the recessive allele is present with the dominant allele, the effect of the recessive allele will be masked by the effect of the dominant allele. When we talk about Mendelian inheritance of monogenic diseases, there is a person whose name will come to our mind automatically. Who is he? He is Gregor Mendel. He performed some interesting experiments using pea plant and established the principles of inheritance. Now let's see how genetic diseases can be inherited through autosomal dominant inheritance pattern. As the name implies, in this case, the mutant allele will be on an autosome and it will be a dominant one. So let's imagine that capital A is the mutant allele and simple a is the normal allele. These alleles can make different combinations and give rise to 3 different genotypes in a population. What are these 3 genotypes? They will be capital A, capital A simple A and simple A genotypes. In this combination, both alleles carry the mutation. So none of them will produce the functional protein. Therefore, the individuals with this genotype will definitely show the disease phenotype. In this combination, what will happen? Here the mutant allele is dominant over the normal allele. Therefore, this mutant allele will not allow the normal allele to express. Only the mutant allele will be expressed in this individual. Therefore, the individuals with this genotype also will definitely show the disease phenotype. Then how about this combination? Simple A combination. This individual is homozygous for the recessive allele and therefore these genes can be expressed. So the individuals with simple A genotype will have the normal or the healthy phenotype. Now let's work out and see the fate of the children if a normal mother with simple A genotype is married to an affected father with capital A simple A genotype. As shown here, during oogenesis, this mother will always produce ova containing simple A or the normal allele. So how about this father? This father will make sperms containing either capital A allele or the simple A allele. Capital A allele is the mutant allele and the simple A allele is the normal allele. As shown by Gregor Mendel, at the time of fertilization, either of these sperms can combine with either of these ova and make these kind of combinations in the genotype of their children. Therefore, if they have a child, there is a 50% chance for this child to have the disease and 50% chance for this child to be a normal one. Here are some examples for genetic diseases which are inherited through autosomal dominant pattern. One is achondroplasia and the other one is amelogenesis imperfecta. The next type of Mendelian inheritance pattern we are going to discuss today is autosomal recessive inheritance pattern. Here also, the mutant allele will be on one of the autosomes, but it will be a recessive allele. That means the normal allele will be dominant over the mutant allele. Therefore, capital A will be the normal allele and simple A will be the mutant allele. These alleles also can make different combinations and give rise to 3 genotypes in a population. What are these 3 genotypes? They are capital A, capital A simple A and simple A genotypes. In autosomal recessive inheritance pattern, capital A genotype will give rise to a healthy phenotype. Then, can you tell me the phenotype of this capital A simple A genotype? It also will be a healthy phenotype. Even though this individual is having a mutant allele, probably he may not even know that he is having a disease-causing allele. But this individual can carry this mutant allele to the next generation. Therefore, he is called a healthy carrier. Then how about this genotype? Simple A genotype. In this genotype, both alleles are mutant alleles. Therefore, none of them will produce a functional protein. Therefore, definitely, this genotype will produce a disease phenotype. In this example, both parents are healthy carriers. That means both of them will have capital A, simple A genotype. As shown here, the oocytes produced by this mother will have either capital A allele or simple A allele. Similarly, the sperms produced by this father also will contain either capital A allele or simple A allele. So at the time of fertilization, these sperms and oocytes can combine in one of these combinations to form a zygote. When a child is born to them, there is a 25% chance for him to be a normal individual and 50% chance for him to be a carrier of the disease and 25% chance for him to have the disease. Some of the genetic diseases which show an autosomal recessive inheritance pattern are thalassemia, sickle cell anemia, albinism and amelogenesis imperfecta. The last type of Mendelian inheritance pattern we are going to discuss today is sex-linked recessive inheritance pattern. As the name implies, here the mutant allele will be on a sex chromosome and it will be recessive to the normal allele. As you all know, we have two types of sex chromosomes. They are the X chromosome and the Y chromosome. Out of them, mostly the X chromosome is associated with diseases. because the Y chromosome is very small and therefore it contains only a very few genes. So rather than calling this inheritance pattern as sex-linked recessive inheritance pattern, we can more precisely call it X-linked recessive inheritance pattern. Here we can see two types of X chromosomes. This is the one with the mutant allele. This is the chromosome with the normal allele. And these are the genotypes and phenotypes we can find in females. And these are the genotypes and phenotypes which are found in males. In females, presence of one X chromosome with a mutant allele will not cause the disease because the normal allele on the other chromosome will be dominant over the mutant allele. But in males, presence of a single X chromosome with a mutant allele is sufficient to cause the disease because there is no normal allele on the Y chromosome to dominate the mutant allele on the X chromosome. In this example, both parents are phenotypically normal. But when you look at the genotype of this mother, you can see that she is a carrier of a X-linked disease. So this mother will produce oocytes with either normal X chromosomes or chromosomes carrying a mutant allele. Since this father is both phenotypically and genotypically normal, he will produce normal sperms containing either X chromosomes or Y chromosomes. At the time of fertilization, these sperms and oocytes can combine in one of these combinations to form a zygote. When a child is born to them, there is a 25% chance for this child to be a normal daughter or 25% chance for this child to be a carrier daughter and 25% chance for this child to be a normal son and 25% chance for this child to be a deceased son. As you can see here, the daughters will be either normal or carriers and the sons will be either normal or deceased. Hemophilia is the well-known example for X-linked recessive inheritance pattern. Another example for this inheritance pattern is amelogenesis imperfecta. Now let's look at another pattern of inheriting genetic diseases which was not described by Gregor Mendel. It is mitochondrial inheritance. As the name implies, the mutation will be on a mitochondrial DNA molecule. Here I have given you two examples. In example A, father is healthy but mother is having a mitochondrial disease. In the example B, father is having a mitochondrial disease but mother is healthy. In the first example, what has happened to the children? Both sons and daughters are having the mitochondrial disease. So what has happened to the children in the second example? Here all the children are healthy. That means when the mother is having a mitochondrial disease, it is inherited to all the children. Both sons and daughters are affected. But when father is having the mitochondrial disease, none of the children are affected. So how can we explain this observation? You can do it if you can remember the process of fertilization. At the time of fertilization, the sperms will shed all its organelles, including mitochondria, and only its nucleus will enter into the ovum. So all the mitochondria in the zygote is derived from the mother. We can analyze the pattern of inheritance of genetic diseases using pedigree charts. A pedigree chart is a diagram that shows the occurrence of phenotypes of organism and its ancestors from one generation to the other. These are the symbols which are used to draw a pedigree chart. Before I finish today's lesson, I want to give you a small activity on inheritance of monogenic diseases. So try to find the answer for having 3 different types of inheritance patterns for the same disease. The name of the disease is amylogenesis imperfecta.